the chromosomal basis of inheritance. When Mendel was doing his studies with pea plants, he'd never seen a chromosome, didn't know what the nucleus looked like, didn't know anything about how these genes were transformed or transmitted from parent to child. We know now, of course, that chromosomes are where the genes have a physical location, and that genes occupy a particular loci or position on the chromosome as the chromosomes undergo segregation and independent assortment. So let's look at independent assortment. This is an example where we've got the yellow round seeds that are heterozygous for, they have the round gene, and they also have the wrinkle gene. They have the yellow gene, and they also have the green gene, so they're heterozygous. And here we have an equal probability that in meiosis 1, the two genes that are dominant will end up in the same cell. So they end up down here. We've got a capital R and a capital Y, so we've got the two dominants together, and over here we have the two recessives together. The small, this will be the wrinkled and the green. All right. It's also equally possible that we'll get the the wrinkled one, which is this small r, with the yellow, or perhaps the round with the green. So each of these are equally probable. And so we call this an independent assortment. Now, genes on the same chromosome do not necessarily assort independently. They tend to be inherited together. So let's look at an example. Here we've got a purple flower and long pollen as what we're looking at. So the purple flower is capital P, and a red flower is lowercase p. The long pollen is a capital L, and the round pollen is a lowercase l. And we expect, if these genes assorted independently, that we would get the 9:3:3 ratio. So the numbers were given here. We get 215 purple long, 71, and 24 of the red round. That's what we would expect. But what we observe is this. There's a high predominance of purple long being together, and for the red round being together. The reason for this is that these genes are linked. They're on the same chromosome, so they tend to be inherited together. When the parent makes gametes, most of the gametes they keep the dominants together and the recessives stay together. And so when we get fertilization, we get the following. We tend to get a dominant here. This would be PL. This would be PL. This is a lowercase p and an L. This is a lowercase p. So this is what we tend to get. And so this individual is PPL. This one is PL. P, little p, big L, little l. This one is lowercase. And this one is homozygous. Genes that are linked can be separated by crossing over. And we can calculate the percentage of recombinant offspring among the total, and this is our recombination frequency. So what happens in meiosis 1 is that the homologs lie really close to each other, and some of them will exchange DNA. And if they exchange DNA between the two loci, then we've separated those genes. And we've made a recombinant offspring. So here we have some crossing over. These two are recombinant, and the others are set. So these are our recombinant offspring. We can use recombination frequency to gauge the distance between two genes on a chromosome. And we call this a genetic map or a linkage map. So we can look at the example we've got here below. We see that G and L have a recombinant frequency of 17%. So we figured that those are farther away from each other. The G and C have a recombination of 9%, so they're closer, and the recombination of C and L is 9.5. So we can look at all the recombinations and kind of figure out where the chromosomes are.